Welcome back, Biology 220. Today we're going to be talking about bacterial diseases of the digestive system. So at the beginning of our digestive system, we have the mouth, where we initially are going to start chewing our food. So our teeth are interacting with a lot of nutrients. And there are over 300 species that have been identified to live on or around our teeth. The most important in terms of causing cavities, or what we call karyogenic. And if it's karyogenic, it means it's caries causing, which just means it can cause cavities or tooth decay. And that bacteria is Streptococcus mutans. So S. mutans, it cannot attach to a clean tooth. So like one of the questions is people have asked, and I looked into recently because I had my first cavity and I started to really consider my dental habits. But one of the questions people have is, should I brush my teeth right when I wake up, or should I brush my teeth after I eat breakfast? And because this bacteria that actually causes the cavities, and we'll look at that process in a second, it cannot attach to a clean tooth. It's better to actually brush your teeth before breakfast, about 30-40 minutes before breakfast, which gives remineralization time for fluoride to interact with your teeth. And it also cleans the bacteria off so that they're not present when you eat breakfast. So then there's going to be less opportunity for cavities to occur. There'll be less acidity from how the cavity ferment those sugars, or how the bacteria ferment those sugars, rather. So within minutes after you brush your teeth, your saliva is going to cover the teeth with a thin film. They call that the pellicle, which is shown here on the right. And it's proteins. And Streptococcus mutans will establish itself on the pellicle. So it takes a little bit of time for that pellicle to form before that bacteria can reattach and reestablish itself. So the bacteria produces this gummy polysaccharide of glucose molecules called dextrin. So it goes through this pathway here, but I think it'd be easier probably just to show you a picture of this pathway, which I believe I have one. Here it is in the top right. So basically what happens is the bacteria feed on the sugars that you eat, and those sugars, after a process and fermentation, are going to release an acid, lactic acid. And that lactic acid damages the surface of your teeth. And eventually, those acids that are produced will start to actually eat away at your teeth. And they will eventually get down into the pulp of your teeth. And as the cavities get deeper and deeper, then eventually they might have to do a root canal, or if it's very shallow, they can just do a filling. And they always grind into your tooth first to grind out the cavity of decayed tissue so they can fill it with amalgam to replace it. So the accumulation of bacteria and dextrin adhering to the tooth make up what they call plaque. So if you don't brush your teeth regularly, then you'll get more plaque because you're not getting that bacteria and dextrin off of your teeth. Now, some of it is going to be left over just by chance. You can't get it all off. And the older calcified deposits will eventually become tartar. We'll look at the difference between those. And that's what they're getting off of your teeth. Now, plaque is generally pretty clear to a slight yellow look. If you look at your teeth, you can probably see some, especially around the edges at the base. And the tartar is going to be a little bit darker, a little bit maybe yellowish to brown versus clear to yellow with the plaque. And at the dentist, when they start to chip at your teeth with those metal prongs, they are knocking off that tartar. Because what happens is the bacteria can be stuck between the tartar and your tooth surface. And as they ferment sugars, then they're going to be producing lactic acid, and that's going to wear down your teeth. So another thing to consider too when avoiding cavities, because of the Streptococcus mutans bacteria, what they do is when we brush our teeth after we eat, you actually should wait about 30 minutes before you brush your teeth if you just ate a meal. And the reason being is right when you're eating the sugary substance, for one, your body's producing lysozymes in the saliva that helps neutralize those acids and sugars. But beyond that, there is going to be acidic substances there. And if you brush them right after you eat, you're actually helping those sugars work their way into your tooth surface. So it's better to wait 30 minutes, let the lysozymes neutralize the acid, let that process go through, and then about 30 minutes later, you can come in and brush your teeth. So this bacteria, they like crevices, areas where there's not a lot of motion and impact. On the top of your teeth from chewing, that makes it harder for the Streptococcus mutans to actually bind to your teeth. So they're going to be looking for those crevices. And that's why you see plaque usually accumulate towards the base of our teeth where there's not that chewing and grinding. So there's usually not very much plaque and tartar where the grinding of our teeth is because it's getting knocked off. Now, saliva does have neutralizing compounds for the lactic acid, but unfortunately, plaque is not permeable to that saliva. So it's not going to be able to actually get through that plaque to be able to neutralize the acids that are produced underneath. That's why it's so important to get routine teeth cleanings so they can get that plaque and tartar off your teeth. Now, as of late, there's controversy about fluoridated water and fluoride toothpaste. Your enamel, if it's low on fluoride, the acid is able to eat into the surface of your teeth more readily. It's more susceptible to the acids. So that's why we have fluoridated water and fluoridated toothpaste, because it helps remineralize the surface of our teeth, the enamel, so that it can protect our teeth from these cavity-causing acids, which come from this bacteria that break down and ferment these sugars that we eat. So it's actually a really good thing. Now, the fluoridated water, the benefit of that is that you have fluoride running over your teeth throughout the day. But really, fluoridated toothpaste is the main thing that I would continue to use if you're against fluoridated water, like the ingestion of it. I understand that, but fluoridated toothpaste is super important. Now, once the tooth decay is started, lactobacillus members enhance the decay through the dentin and the pulp. So as it continues, it's going to get deeper and deeper. So how can you avoid cavities? Well, we talked about breakfast time. You should brush your teeth first thing when you wake up with fluoridated toothpaste. That's going to get the bacteria off your teeth. It's going to get that pellicle off so they can't attach. And you're actually going to have about 2 hours where that bacteria is not going to be in your mouth at a high population density. So brush your teeth right when you wake up, get a shower, whatever, give yourself about 30, 40 minutes, then eat breakfast. And then it's not going to be damaging your teeth. You don't want to eat breakfast and then brush your teeth right after because, again, you're grinding those acids into your teeth. So you don't want to do that. If you're going to drink like a sugary Gatorade or drink a soda, excuse me, always do that with your meals because the lysozymes in your saliva can neutralize those acids, but that's going to be worn out after your meal. So if you eat sugar between meals, like you like to snack on sweet things and muffins and Twinkies, well, it's between the meals where you're going to get damage to your teeth. So obviously limiting how much sugar you eat is a great way to avoid this because then you're going to avoid that sugar becoming an acid from that bacteria interacting with it and breaking it down. So avoid sugar. Also, like things you don't think about that are also sugars, like bread, tortilla chips, anything that has carbs in it is sugar, and it does the same thing. So you may eat, a piece of bread in between meals, and that's just as bad as eating a cookie because it's a ton of sugar for that bacteria to ferment. Obviously, you want to regularly clean your teeth and get professional cleanings. Fluoride helps remineralization of the enamel. And then chlorhexidine mouthwashes. Mouthwash is great, fluoridated mouthwash. And this kind, chlorhexidine, helps, it's antibacterial. So it kills the bacteria that causes cavities, which is a huge benefit. So we'll look at some different diseases here now. So our first disease is periodontal disease. So this is a number of conditions characterized by inflammation and degeneration of the structures that support the teeth. So our gums and our jawbone. So gingivitis would be inflammation of the gums. So if someone has gingivitis, because it's inflamed when they start to floss or brush their teeth, it'll bleed. And that can be from this inflammation of the gums. Periodontitis, this would be a chronic condition that results from the progression of gingivitis, which is responsible for 1/10 of all tooth loss in adults. And I'll show you some pictures of these. It can get to the point that we get something called acute necrotizing ulcerative gingivitis, or what they call trench mouth. I think the notes, or at least one thing I saw, it said it was common. It's not common. Trench mouth is not a common condition, but gingivitis is common. But trench mouth is much more serious and much more rare. Trench mouth can a contributing factor to that can be high levels of stress, which actually compromises your immune system and can lead to trench mouth. So here's some pictures of this. In the top left picture, that's inflammation of the gums. You see where it's nice and red. So that would be gingivitis. In the top right, we have periodontitis. So that's, you see that pus pocket there that arrow is pointing to would be periodontitis. And then here it just shows you the degradation or the progression of periodontal disease. The gums start to get inflamed, they wear down, they separate from the tooth. You'll notice where that plaque builds up just gets more and deeper, which means you're going to have deeper and deeper tooth decay and cavities on your teeth, which can lead to them having to be pulled because down here you can't just do a filling to fill that in. And then there in the bottom right, you have where it gets to trench mouth. So some lower digestive system terms that we'll look at. We see the term just infection. So that's when you get a pathogen in there and it starts to grow. So it's characterized by delayed in the appearance of symptoms, often accompanied by a fever, versus intoxication, where you have exposure to a toxin, ingestion of a toxin, and that's going to be more sudden. So infections might take time for it to grow. Intoxication can be more sudden, have sudden occurrence. So both can result in the same thing: diarrhea, vomiting, abdominal cramps, nausea. We'll look at tons of examples of this. You probably had some examples of this from food poisoning. And then if it's severe diarrhea and there's blood and mucus in it, we call that dysentery. And then gastroenteritis would be diseases causing inflammation of the stomach and intestinal mucosa. So we'll look at some examples. Staphylococcal enterotoxicosis. This would be food poisoning is the name, or it's what this is. So this would be a major type of food poisoning in the United States. It is an intoxication caused by ingesting an enterotoxin produced by the gram-positive Staphylococcus aureus. So the microbes enter the food, they incubate. They call this temperature abuse. That would be when there's like improper handling of the food that allows it to remain in what they call the temperature danger zone. And what that is it's temperature between about 41 and 135 degrees Fahrenheit, or 5 to 57 degrees Celsius, that bacteria love, right? When we incubate our bacteria, when we're trying to make them grow, we're within that range. So if food's left out in that range, well, then you're allowing the bacteria to grow. So they reproduce, and then they can release toxins into the food that then we eat it, it makes us sick. So what can happen is someone can actually cook the food properly, but because it was left out so long, it produced toxins that are still going to be there after you cook the food. The temperature may kill the bacteria, but that doesn't mean it killed or destroyed the toxins. Those are still going to be there, and that's still going to make people sick. 1 million bacteria per gram of food is enough enterotoxin to cause illness, which 1 million bacteria is a very small amount, actually, compared to, like if you remember, we did bacterial ubiquity, and we calculate how many bacteria fit on a slide, a single glass slide in the lab. Remember, it was billions. So there's going to be, that's really not that much, especially if it's been left out for a while. So staphylococci, they're resistant to environmental stresses such as heat, radiation, osmotic pressure, and desiccation. So they're very resilient little guys, which can make it more prone to causing foodborne illness or food poisoning. So what this process looks like is the food containing protein is cooked, the bacteria may even be killed, then the food is contaminated by a worker. Maybe they don't wash their hands after going to the bathroom, or they've just touched some surfaces that had the bacteria on it. They prepare the food is left out at that room temperature, in that temperature danger zone. The organisms incubate and grow, release toxins, and then maybe the food is reheated even, but the toxins aren't going to be destroyed from that. And then the food containing those toxins is eaten by a customer, and then in about 1 to 6 hours, they're going to start having those symptoms, which you probably had before if you ate at a restaurant and then a couple hours later, you have diarrhea or you're puking your guts out. So if you want to avoid this, now going out to eat at a restaurant, you're not really in control of this, but in your own food preparation, obviously washing your hands is going to be one of the best things you can do. And then, of course, refrigerating your food as soon as you're done with it. Like you're done with dinner, refrigerate your food. That way it's not left out in that temperature danger zone. So there's a really cool, I shouldn't say cool, but a really interesting story. You may have heard of this. This was in 1993. There's a restaurant called Jack in the Box. You'll notice in the map up here, it's not really around us anywhere. But Tennessee, South Carolina, Texas, and California is where you're going to see a lot more of these stores. But it's a restaurant that has a wide variety of food types. Right, you'll see here in the bottom right, they've got tacos on their billboard, and of course they also serve hamburgers. So anywhere that serves both of those might be a little bit suspect. But this actually wasn't their fault at all. They were ordering patties from a particular company called the Vaughn Company. And the patties they ordered from there that they were preparing properly were, they found out later after an investigation, that they were infected with E. coli, a particular type, O157H7. And it's a very dangerous strain of E. coli, and it causes something called hemolytic uremic syndrome. And basically what it does is it creates the destruction of red blood cells. It can lower your platelet count, and it can damage your kidneys. And it can be really dangerous. It actually had 4 confirmed deaths from these patties from the Vaughn Company. There were over 700 people who got sick. 171 went to the hospital for several days, and then 4 people ended up passing away from that Jack in the Box E. coli outbreak. And that wasn't even their fault. That was just from where they got the patties from. So Shigella, or bacillary dysentery, this is caused by members of Shigella, a facultatively anaerobic gram-negative rod. You see there in the right, gram-negative, that pinkish red color, and then the rod shape, bacillus. Infection is usually fecal to oral route. You'll see that with a lot of these foodborne illnesses. So the residents of the intestinal tracts of humans, apes, and monkeys only. So when you're getting this, unless you had a literal animal prepare your food or someone touched one, you're probably getting it from humans who did not wash their hands after going to the bathroom. And then when you get it, you're going to experience very intense watery diarrhea. And the main threat with most cases of foodborne illness or diarrhea is going to be the threat of dehydration. So some people who get diarrhea when they travel to other countries, it could be shigellosis. So for salmonella or salmonellosis, this is a gram-negative, facultatively anaerobic, non-endospore forming rod. For this one, there's a little bit of a delay. It might be 2 days later before you actually have these symptoms. And once they start, it might last a day to a day and a half. And you're going to have a fever, nausea, and diarrhea. So the source is usually the intestinal tract of animals, and we get it from foods that have that bacteria on it. A lot of times people get this from raw eggs and chicken. So you got to be careful. Like if some chicken was left out all day, even if it was from the grocery store, but then you left it in your car all day, you still wouldn't want to eat it. And the FDA recommends that if chicken is left out for more than 2 hours, or 1 hour if it's above 90 degrees Fahrenheit, you should not eat it, which seems ridiculous, but you're putting in that temperature danger zone for 2 to 4 hours is actually a really long time for that bacteria. So then it's getting to the point where it can make you sick. There's high enough concentration of it. This can also contaminate inanimate objects, so fomites, like cutting boards. Wooden cutting boards can be a little bit sketchy because you're cutting into it and it's making grinds and increasing the surface area of the cutting board, and then you leave the raw chicken on there perhaps, and you might wash it off with soap, but you don't scrub it very well. You might not have gotten it all off of the wooden cutting board. So you have to be careful with that. You want to clean them really, really well. or just use a different cutting board for your raw meats that you know you're going to be cooking right after than you would with your fresh fruits and vegetables. Because you cut the raw meat on it and then you cut vegetables on it, and now you've cross-contaminated. So there's a lot more cases of this than actual hospitalizations, excuse me, or reports, but nonetheless, it's still pretty common. So another disease we have is Salmonella typhi. This is the most virulent of the Salmonella serotypes, spread only by the feces of humans, and it incubates for about 2 weeks. High fever, continual headache, diarrhea appears during week 2 and 3 after the fever goes away. Now, all of these diseases, with proper hygienic practices, you're really not going to get these. So if people just wash their hands properly before they prepare food and clean surfaces very well, like in a kitchen, at a restaurant, or in your home, you're not going to be spreading these. Now, what's really interesting about this, about Salmonella typhi, is that recovered patients can actually become chronic carriers where the bacteria lives in their gallbladder and is shed in their feces for maybe months, years, or forever. So someone could not feel sick from this anymore and actually still have it for the rest of their life. And an interesting example of this that's very famous is called Typhoid Mary, where she recovered from typhoid fever and she felt fine, and she was preparing food for people and was causing typhoid fever in all these different families. And we'll actually look at that here in a second. But again, you're going to get this from contaminated food. For those who are carriers, can have this collected in their gallbladder, and then they'll pass it on potentially indefinitely. So for Typhoid Mary, just kind of basing this off my memory, and I don't want to, I'm sure I'll make a couple of generalizations here, so I apologize for any inaccuracies. But basically, the story was she was a cook, and she was preparing food for all these different wealthy families. She was a cook for 8 different families. 7 of those 8 families were claimed, at least, if not confirmed, to have had typhoid fever around the time that she worked with them. And when she would go to a different wealthy family to be their cook, she would never, of course, tell anybody where she was going. She would come quickly and leave quickly, and she would deny, because there was a point where they were investigating, and people were figuring out that actually she's the common denominator between all these different families and people who have had typhoid fever. And anyway, long story short, they found out that they tested her for this bacteria, and they found that it was actually in her feces and urine, and it was residing in her gallbladder. So here's a little excerpt of what happened. So it said for 4 days, she was not allowed to get up and use the bathroom on her own. So she was actually quarantined, ended up being quarantined twice. The first time, to figure out what was going on, she went back to cooking for people and getting them sick, and they had to quarantine her indefinitely after that. But the first time, they found massive numbers of typhoid bacteria in her stool samples, and the infection source was her gallbladder. Here's some pictures of her to the right. Top right, she's cooking for someone. Bottom right, that's her in quarantine. So during questioning, Malin admitted that she almost never washed her hands. And we think, what are you doing? Like, you're a cook for a family, why aren't you washing your hands? And again, it explains here, but people weren't thinking about germs back then. This was in the late 1800s, the early 1900s. I think it was 1907 when she was quarantined. People weren't thinking about that. That was very common to not wash your hands before and after preparing food. That brings us to cholera. So for cholera, this is a slightly curved vibrio is the shape with gram-negative rod. So you see it here in the top right. It's got a slight curve to it. Some of these you see a little bit more. It's gram-negative. Causes cholera, which can be fatal. With proper treatment, it won't be. There's about a 1% fatality if they're treated properly. But if they're not, then there's a very good chance that they can actually die from it. And what it does is it comes from contaminated seafood, foods that have been in salty brackish water. Animals that are in brackish water will become infected and transmit this. So undercooked seafood would be a very common source of this. And one of the ways to tell this from other diseases is that because it sheds water and a lot of minerals in the stool, it creates this white water appearance, like a rice water appearance. And we'll look at some pictures of that actually to show you what that looks like. It could cause you to shed up to 20 liters of fluid in a day, which to gallons, that would be about 5 gallons. 20 liters is about 5 gallons. So 5 gallons of milk, imagine that coming out of your body in one day. So you can see why it could cause someone to die of dehydration. So here we've got a couple of examples of different stools. So on the left, green watery stool. So that might be from rotavirus. On the right, we have the rice water description of stool from cholera. So you can see it looks like rice in water. And that's all those minerals and calcium that's in there that makes it look white. So Vibrio parahemolyticus gastroenteritis. This would be non-cholera gastroenteritis. Also found in saltwater estuaries. And this comes from eating raw oysters. So every now and then there'll be some kind of like public service announcement about for people who are traveling to other countries about different types of foodborne illnesses to watch out for because they've had some type of outbreak in those areas. So for example, this one is about South Australia. This bacteria is in raw oysters. So it says don't eat raw oysters when you go to South Australia. So you'll see little things like that associated with these foodborne illnesses. So E. coli, this is probably the one that everyone knows of. They also call this E. coli gastroenteritis Montezuma's revenge. We'll kind of look at the story for why that's the case. But this is one of the number one causes of travelers' diarrhea. And again, it's a gram-negative rod. We've already looked at these before. So these have specialized fimbriae that allow them to bind to certain intestinal epithelial cells, and they produce these toxins that cause diarrhea. So you ingest contaminated food that has fecal material on it, which is where E. coli comes from the intestines. We have E. coli in our intestines right now. Now, they call it Montezuma's revenge, and it's given that name because it's often experienced by travelers who go to Mexico and Central America. And you think about Montezuma, he was the last Aztec emperor who supposedly plotted revenge against invaders by by giving gifts that were wrapped in fecal, that had these fecal coliforms on it, this E. coli, that would give travelers diarrhea. So it's kind of the reason behind that name. So from this, you get it from undercooked meat, raw milk. If fecal material were to get on fruits and vegetables and someone were to eat those, then they could get it. And it could cause diarrhea, fever, vomiting. And then we see that hemolytic uremic syndrome as well. So death rate, pretty low, about 3 to 5% for that particular type. Then we have Campylobacter jejuni, which is gastroenteritis, gram-negative, microaerophilic, so it needs lower levels of oxygen, and it's a spiral curved bacterium that we see here on the top right. So this is a cause of foodborne illness. It lives in the intestines and especially in poultry. So in poultry and chicken. Second common cause of diarrhea, staph is the first. And usually it's not going to be life-threatening. Jejuni, because if we think about the jejunum, that is in the small intestines, and it's that middle section of the small intestines between the duodenum and the ileum, and it plays a crucial role in nutrient absorption, particularly of sugars, amino acids, and vitamins. So it's got this thick mucosal lining, numerous villi, and a rich blood supply, and that's where, for all of those reasons, this bacteria likes to reside. So you can pause the video and look at some of these sources and symptoms. One of the more interesting ones is it can cause hepatitis or inflammation of the liver, inflammation of the pancreas, and some other parts of our body like our joints. Helicobacter pylori. For this one, this is a peptic ulcer disease. And the story behind this is actually really interesting. But it's a spiral-shaped bacterium, and it causes ulcers in the stomach. And for the longest time, People thought ulcers were caused by eating acidic foods and stress, which can contribute to ulcers, which could be a cause of ulcers. But it can also be caused by this particular bacteria. And when they got down to figuring out like that this was causing ulcers, two researchers that were studying this, they were pretty sure that this bacteria was causing these ulcers. And they were able to find this bacteria in the stomachs of people who had ulcers. But to prove that this was causing ulcers, they had to test it. They had to test their theory to confirm it for sure. So this little excerpt here I'll read you. It says, Dr. Barry J. Marshall was convinced that Helicobacter pylori bacteria caused stomach ulcers, but since no one believed him and he couldn't just test this on somebody, he couldn't just give someone this bacteria to make them sick, he ingested the bacteria himself, developed the ulcers, treated himself with antibiotics, and they went away, which proved that Helicobacter pylori was causing stomach ulcers. So very brave that he did that, and he won the Nobel Prize for it, which is pretty neat. So Yersinia enterocolitica, this is a gastroenteritis transmitted through meat and milk, and it can grow at refrigerator temperatures. And this one causes severe abdominal pain that is often misdiagnosed as appendicitis. And I got food poisoning once from a place in, I forget what it's called now, Lambert's. You know, they have the huge rolls and the great food. And anyway, I thought I had appendicitis because where the pain was, but it turned out to be food poisoning. But it's often misdiagnosed as appendicitis because it hurts right in that same area of your stomach where when your appendix gets inflamed, you also feel pain in that area. So Clostridium perfringens, gastroenteritis, large gram-positive endospore-forming obligately anaerobic rod associated with meats and meat stews contaminated during slaughter. The exotoxin caused the abdominal pain and diarrhea. Symptoms appear 8 to 12 hours after ingestion. So usually you're going to experience food poisoning. It might be very quickly, like right after the meal, within an hour to 6 hours, like within the day. And some will actually take weeks of incubation, but usually you'll experience it within the same day or the next morning. Our next one is Clostridium, and the way you say this, it's not difficile, it's difficile in the way the French, which is where we get that, the pronunciation is, or the Latin way I've heard is difficile, but usually people say difficile in common English, even though it comes from a French word. But for short, to make it easy, you can just say C. diff. So this causes inflammation accompanied by increased fluid secretions of the intestines. This will be nosocomial, which means it comes from hospitals and nursing homes where it could be acquired. So if someone uses a broad spectrum antibiotics that disrupt the normal flora of the intestines, then this bacteria can come in and take up residence there. It has a distinctive odor, serious colitis, inflammation of the colon, and it can cause death. When people fight off this infection, then they can take transplants, which is literally getting the healthy bacteria like probiotics to try to reestablish a normal flora so it doesn't happen again. So again, when people take antibiotics, especially ones that kill a large variety of different types of bacteria, then it kills the good bacteria and the bad bacteria like C. diff, which has these endospores, which make it through the low pH of the stomach, can then get into the large intestines and the small intestines and start to develop. For this bacteria, it's pronounced Bacillus cereus, and you can remember this one because it's very serious, because when you heat it up, it doesn't kill the spores. So the spores can still survive because nobody's cooking their dinner in an autoclave. So the spores still make it through. When you heat this up, when you heat these foods that have this bacteria in it, it might kill the competing bacteria, but then this bacteria can grow back and replace it and then it starts to produce toxins. So where you might get food poisoning from an Asian restaurant is if they try to reuse the rice from previous day to the next day, and even though it had been heated before they refrigerated it, the spores can still germinate, and then when they start to reheat it the next day, and then that bacteria starts to develop, and towards that afternoon, everyone who's eaten the old fried rice is now getting sick. So here's some other information on Bacillus cereus if you want to pause the video and look at some of the tests and treatments and how they diagnose it. So just last thing here would be coliform, or the coliform test, and how they test for water purity. So this would be the most commonly used indicator for water contamination to see if feces has gotten into the water. So this would include all gram-negative rods, aerobic and facultative is what the coliforms are, what we're testing with coliforms. Those that ferment lactose and those that produce gas. Includes organisms associated with the intestinal tract. So that would be fecal material. If feces had gotten into the water, then it's going to show up on a coliform test. And I'll show you some different tests that I actually have done in the past. Now, just one little fact before we move on to the next slide is in countries that don't have like proper sewage treatment where a lot of feces and human feces gets into the water, in third world countries, there is an average, let me see this fact here, over 12,000 children die every day from diseases caused by waterborne fecal pollutants, which is pretty crazy to think about just because they don't have clean water. And it's because feces is getting into the water from animals or humans that is contaminating the water. So you'll see there that this other type of tube test for coliforms. What I did was I was an Alabama Water Watch citizen science volunteer, and our science club would go out and we would test water for coliforms. And we have these things called R cards, which are made by a company called Roth Biosciences. And I would encourage anybody, especially science teachers, if you're interested, to get involved with Alabama Water Watch, because it's just a great program to keep an eye on our waterways and make sure that they're staying safe for people to swim in, fish in, and even like, could you drink out of this water? And basically the way our card works is you put a milliliter of a water sample from the creek or river or whatever you're testing, a lake, you put it in the R card, you close it, it spreads it out, and then you put it in an incubator. So it's basically like a growth plate, but it's just a flat, simple card. And they cost about $2 to $3 a card. Depending on how many you buy, you can get a discount for quantity. So where I tested, I was teaching at Raglan, so we would test Trout Creek right behind the school. And you see that's actually our mark right there. You can look up Alabama Water Watch data map, and you can go all over the state, and you'll see all these red and green dots. Red means it's no longer active, green is still active. And you can actually go to Raglan and look at all of our data and our tests. And I'll show you some tests that we did here in just a second. But the way you read the chart is basically if something's blue, that's fecal coliforms. That's E. coli that's going to make you sick. Other non-pathogenic coliforms show up in red and pink. So red and pink is good, blue is bad. So if you look at this card, you've got a lot of blue. So those are all caused from one E. coli bacterium. Each one of those blue dots is one E. coli bacterium that is now reproducing asexually. And that's in 1 milliliter. So right here you got 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, maybe 17 or 18. Depends on if we count that one. So then in 100 milliliters, you would multiply that by 100. So there'd be 1,800 fecal coliforms per milliliter, which is a lot of E. coli. So this was a chart one of my students report that I helped them write based on these studies that we were doing, these tests. So we would go out, we would measure the water before a storm, and then we would measure the water after a storm. And if you look, before a storm, there were barely any E. coli colony forming units, or those purple dots. But then after it would rain, and these are two different tests that we did twice, after the storms, you'll notice it went up above 1,000. And I have a chart for what is a safe level here. Yes, So the upper limit, it looks like, is 600, and we were at well over 1,000 colony forming units. So literally the water in Raglan, after it rains, like a storm comes through, is according to like the CDC, according to any like EPA, you should not touch that water. It is not safe for human contact. And people in Raglan would always swim in the water because it would flood and it would be fun. And I would ask them, I'd be like, well, do y'all get like pink eye, ear infections or anything when you swim in the water? And they're like, oh, yeah, we get ear infections all the time. So, yeah, because you're swimming in feces, like E. coli is everywhere. And Raglan's basically like a bowl that funnels into Trout Creek. And there's a lot of cows and chicken houses. So all that feces just runs into the water. And because it's shaded during a storm, there's no UV radiation to kill the bacteria. So it just grows and reproduces, especially in the summer when it's warm and people would get infections from it. So after it rains is actually a really bad time to go swim in creeks, especially in rural areas where fecal material is going to get down into those creeks. So I put this up here just to say, in conclusion, the best thing, like for example with polio, one of the best things that decreased the incidence of polio was, of course, vaccines, but also just proper sewage and plumbing. If you can just get the poop away from people, you're going to cut down diseases by 99%. Vaccines help a ton too as well, but the most important thing is keeping people from coming in contact to feces in our food, in our water, and those types of things. Sure, it drastically cuts down on disease. And I think that has a greater effect than anything else we've done in modern history, is getting people away from the contaminants. And then, of course, all the other stuff is good too.